Today we are going to start a new topic in microbiology, that is Salmonella. It is a kind of bacteria. Salmonella are of two types. The first is Salmonella typhi and the other one is Salmonella paratyphi. There are also other species of Salmonella, but these are main. Now coming on some diseases that are caused by Salmonella. Some of them are enteric fever, gastroenteritis, septicemia. What is enteric fever? Enteric fever is also called as typhoid fever. It is caused due to intake of contaminated food which is contaminated by this bacteria. It causes typhoid fever. The Salmonella typhi causes typhoid fever and the Salmonella paratyphi causes paratyphoid fever. Typhoid is accompanied by a high degree fever for 2 to 3 weeks. For 2 to 3 weeks. Okay. Now coming to gastroenteritis. Gastroenteritis is also called as stomach flu. These are the species of Salmonella that are responsible for gastroenteritis. It is caused due to intake of contaminated food which is contaminated by these types of Salmonella. The other one is septicemia. Septicemia is blood poisoning which is also caused due to Salmonella. Now coming on morphological structure of Salmonella. It is gram negative in nature. The dimension of Salmonella is 1 to 3 micrometer into 0.5 micrometer. These are motile, that is peritrichous. What are peritrichous? When the flagella is present all over the cell body of a bacteria, it is called as peritrichous. What peritrichous? Peri means periphery. Okay. There are some exceptions. These are Salmonella gallinarum and Salmonella pylorum. They can't move. They are non-motile. They are not peritrichous. They are non-motile. These bacteria are non-sporing and non-capsulated also. All Salmonellas are non-sporing and non-capsulated. Now coming on culture of Salmonella. Culture means the way these bacteria grows. Okay, on a particular medium. So basically, Salmonella are non-fastidious. What's non-fastidious? It means it do not need any kind of a special conditions or requirements for its growth. It could simply grow on an ordinary culture. Okay, there is no need of special conditions. The temperature needed is 37 degrees Celsius and the pH necessary is 6 to 8. Salmonellas are aerobic and facultative anaerobic. Aerobic are those organisms that could grow or live in presence of oxygen, and facultative anaerobes are those organisms that could live and grow both in presence and absence of oxygen. So Salmonella are aerobic and facultative anaerobic. They could grow even in the absence of oxygen also. When these bacteria grows, they form colonies. What are colonies? It is a group of bacteria that accumulate to form a large colonies of that particular bacteria. Okay, so the colonies of Salmonellas are circular. It is 2 to 3 mm in diameter. It is low convex, translucent, and smooth. So these are some characteristics of the colonies of Salmonella. Okay, now coming on the culture medium in which Salmonella can grow. The first one is on McConkey agar. On McConkey agar and on deoxycholate citrate agar, the Salmonella produce colorless colony. In these two, the Salmonella produce colorless colony. Why? Due to non-lactose formation. So here on these two medium, no lactose is formed, due to which the colonies remain colorless. Okay, you have to learn this. The second type of medium is Wilson and Blair bismuth sulfite medium. It is a selective medium. So, what's a selective medium? See, when we have to isolate a particular bacteria or a particular species of bacteria from a group of number of bacteria, we have to use a selective medium that could select that particular bacteria we want. So in this way, this is the selective medium for Salmonella. Okay. So when we grow a particular group of different kinds of bacteria on this medium, okay, so the area where Salmonellas are present, there forms jet black color colonies. Okay. Why? Why these jet black color colonies are formed by Salmonella? It is due to formation of hydrogen sulfide, H2S. Okay. So this is the way we can select Salmonella from a group of number of bacteria. Understood? And on that same medium, the Salmonella paratyphi and other species produce green colonies. Other species of Salmonella only, okay? They produce green colonies because they don't form H2S. So two kinds of colonies will be formed, that are jet black colored and green color colonies. The second medium is xylose lysine deoxycholate agar. It is a different selective medium which is also used for selection of Salmonellas. In this medium, most of the strains of Salmonella produce red colonies with black centers, as it is seen in this image. And the Salmonella which are H2S negative, that is those who don't produce H2S or hydrogen sulfide, they produce red colonies but with no center, no black center. Okay, so this is the difference. Selenite F broth and tetrathionate broth, TTB, these are enrichment medium that are used for inoculation of specimens, especially feces. Okay, so here what is the enrichment medium? So enrichment medium is that medium which has different kinds of nutrients and required substances that are required for growth of the particular species or the growth of a specimen. Coming on some biochemical reactions that are carried out by Salmonellas. These bacteria ferment glucose, maltose, and mannitol to produce acid and gas. There is one exception, that is Salmonella typhi, which ferment all these. Salmonella typhi also ferment all these, but as a product it produces only acid. No gas is formed by Salmonella typhi during the fermentation of all these. Okay, this is the difference. Lactose and sucrose is not get fermented by any type of Salmonella. The another biochemical reaction is most Salmonella produce hydrogen sulfide in triple sugar iron agar. In this particular medium, which is triple sugar iron agar TSI, most of the Salmonella produce H2S, hydrogen sulfide, while growing, okay, except these two. The another biochemical reaction is they utilize citrate except Salmonella typhi and Salmonella paratyphi. These Salmonellas are MR positive. What is MR positive? Whenever the glucose is fermented by Salmonella, the fermentation of glucose produce acidic medium up to 4.4 or below it. Fermentation of glucose produce acidic medium, due to which the culture media turns red. That's why they are called as MR positive. Resistance. Salmonella can be killed at 60 degrees Celsius in 15 minutes. Chlorination of water could also kill these bacteria. Pasteurization of milk, that is boiling of milk, kills these bacteria. Mercury chloride and phenol also kills these bacteria. The salmonella can survive in water, ice, and snow for months. Okay, so this is the resistance of salmonella for months. So this is enough for today. If you like this video, please do like, share, and subscribe. Thank you.